In this video, I'm going to teach you how to use a microscope in the lab to look at your animal and plant cells. When moving the microscope, if you should ever have to move it, you're going to make sure that it's unplugged and that you're carrying it with one hand on the neck and one hand on the base. So you should always have two hands on the microscope, one on the neck, the other under the base. But you shouldn't have to carry the microscope at any time. Just a quick review of the parts of the microscope. The top is the eyepiece. This is where you look into. These are your different objective lenses. They show you the magnification, how you're looking at the specimen. This right here is your stage. Your stage is where you place the specimen. You want to make sure that your specimen is centered right over the light. These are your stage clips. You lift them to secure the specimen, the slide in place. This is your focus adjustment knob. You turn this to adjust your focus by lifting and lowering the stage. This is your light switch. Light must be on for you to observe the specimen. You can rotate the eyepiece in order to look from different angles. You always want to make sure that your microscope is plugged in, especially if the light is not on. This one is charged, so it's okay. Before we begin learning how to use the microscope, I'm going to teach you how to make what's called a wet mount slide, which is what you'll use to look at your cheek cells. So you want to start by getting one glass microscope slide. You're going to take a toothpick to scrape the side of your cheek. So we'll scrape the side of our cheek with the toothpick. Then you're going to take one drop of water, place directly in the center, like that. You're going to tap the tip of your toothpick, the cells that you acquired from your cheek. You're going to release them into the water. And then on top of that, you're going to add one drop of iodine. For this case, I'm just using red food coloring. You will use iodine. And then you're going to take the slide cover, which looks like this little square. You're going to put it at an angle. You notice it's at an angle, and carefully drop. Just like that. Now you have a prepared slide. Again, yours won't be red. This is just an example. At this point, you will carefully place the slide underneath these stage clips. So you'll lift each stage clip and you'll put the slide underneath. To look at your cells, you want to make sure that the light is on. This is the on-off button. You want to make sure that the stage is all the way lowered by using this large knob here, pushing it forward. That's how you lower the stage. And you also want to make sure that your objective lens is at the shortest or smallest magnification. So the red. You're going to start by looking in to the eyepiece, and then you're going to slowly adjust your magnification until something comes into focus. You won't be able to see, but that's what you'll do. Once you get it focused, then you're going to very carefully turn the objective lens to the next one 10 times. Do the same thing, focus it using the focus until it comes into focus. If you can get that clear, then you may move to the highest level. Now in this stage you have to be very careful, because if you notice, this is a longer lens, and so if it comes in contact with the microscope slide, it could break it. So you don't want to break the microscope slide or you will not be allowed to do the lab anymore. So make sure that you're being really careful when you're on the highest objective lens. Start with the lowest, which is the red. Move to the middle. If you get that cleared, make sure you ask me before you move on to the high objective lens. When using the microscope, you want to make sure that it's always a hand width away from the edge. So you can't bring it any closer to the edge than about right there. You also want to make sure that the light is on and that it's plugged in. You also want to make sure before you begin that the stage is all the way down. And before you begin, you should also make sure that you're on the lowest objective level. Let's talk real quick about how to make a dry mount slide. When you're looking at plant cells or your onion, you'll make what's called a dry mount slide. You'll start by grabbing one slide. You want to make sure it's clear from dust. This one is not. You'll want to clear yours off. You will set your specimen or small slice of plant or onion right on top in the center. This is gecko skin. I'm going to make a dry mount with gecko skin. And then you're just going to grab another slide. Excuse me. You're going to grab another slide and you're going to place it right on top. Very carefully right on top. That's all you do to make a dry mount slide. Okay? Then you're going to carefully pick up your dry mount slide. Yours should be even. It's hard to do this with one hand. And you're going to place it under the stage clips, just as you did your wet mount. Focus it just the same to look at the plant cells. Wet mount slide, dry mount slide. Hopefully now you have a good understanding of how to use the microscope. Get excited to look at some specimens.